Hello and welcome everyone to this video podcast for evolution and diversity. In this podcast, we're going to talk about patterns of natural selection. I'm going to open with a topic I should have put on the last podcast, but I want to make sure that you have it. So macro versus micro evolution. And then we'll move into some of these patterns. We're going to talk about artificial selection. We're going to talk about 4 different modes of selection that affects the genetic stability and variation of a population. We'll talk a little bit about sexual selection. All right, so let's go ahead and get started with our first topic of macro versus micro evolution. All right, so let's compare micro and macro evolution. We think about micro evolution being on a smaller scale. What I mean by that is it occurs over a shorter time period. Say over the course of our lifetime, we will see evidence of microevolution. It occurs in single populations. So some examples of microevolution would be things like antibiotic-resistant bacteria. So for instance, if someone doesn't take all of their antibiotics that they're supposed to take, sometimes that bacteria will adapt to that antibiotic and become resistant to it. An example of microevolution. Same thing with herbicide-resistant crops. Crops can become herbicide resistant. And the last example is HIV resistance. We know that there are small populations in the world that are resistant to HIV, meaning if they're exposed to HIV, they don't come down with symptoms related to HIV or AIDS. And this topic may come up a little bit later in the semester. But these are all 3 examples of microevolution, things that we can see in our lifetime. Macroevolution, as you might guess, is on a larger scale. It occurs over a longer period of time, maybe even millions of years. We would say that it transcends a single population of a single species. Some examples of macroevolution would include the origin of eukaryotes. That didn't happen over a shorter period of time. That took millions of years to develop into a eukaryotic system. Origin of vertebrates. Macroevolution also includes extinction. So we could think of the extinction of the dinosaurs. Lots of examples of macroevolution. And as we move forward in this course and we start talking about the diversity of everything we see on Earth, we're going to talk more about events of macroevolution. It's important to remember, though, that microevolution and macroevolution, they both rely on the same mechanisms of evolutionary change. That is, things like mutations, migrations, genetic drift, and natural selection will influence whether or not micro or macro evolution occurs. Let's move on to artificial selection. Let's begin with a definition of artificial selection. It is the deliberate manipulation by humans of the genetic composition of a population by allowing only individuals with desirable traits to reproduce. Turns out that Darwin took advantage of artificial selection when he used pigeons as a model system. He did this to study how the characteristics of a particular population changes over time. And this helped him establish some of his ideas about natural selection. I always think it's interesting that in this time period of the 1850s and 1860s, there was another scientist that was also making his mark in history, and that was Mendel, Gregor Mendel. and you'll talk more about Mendel when you take genetics, but just real quick, he was studying essentially the genetic inheritance of traits. Now, even though they were both making their marks in history at about the same time, there's really no evidence at all that they ever corresponded with each other or ever read each other's works. But one could imagine that if they did actually meet and share their works with each other, there would have been a lot of synergism between their findings. Mendel would have provided a genetic explanation to what we see in Darwin's predictions. But sadly, they never interacted with each other. Okay, let's go ahead and move on to the next objective, and that is to discuss modes of selection and how that affects genetic variation and genetic stability in a population. When we think about these modes, there are 4 that we need to consider. The first is directional selection. The second is stabilizing selection. The third is disruptive selection. And then our last one is balancing selection. I'd like to give a brief definition of each of these, and then I want to go on and talk about each one in a little bit more detail. Directional selection changes the average phenotype in the population in one direction. Stabilizing selection does not change average value of a trait or time, but reduces the number of individuals at both extremes. Disruptive selection selects against intermediate phenotypes and favors the extreme phenotypes. Balancing selection occurs when there is a balance among alleles in terms of their fitness and frequency. Okay, so now let me go over each of these 4 in a little bit more detail. Okay, so let's talk about directional selection. And as we talk about each of these selection patterns or modes, we're going to refer to a very common graph, where on the vertical axis we have the number of individuals. Along the horizontal axis, we think of this as the traits, or the phenotypes we might say. Or we could also think, and maybe more appropriately think of this as allele frequencies. The number of individuals in that population with a particular trait, which is linked to the allele frequency. So what we would see before selection occurs is a fairly normal distribution of traits. So for instance, we might have on this side a dark pigmented mouse. On this side, maybe we have a light pigmented mouse. And in the middle, we have a bunch of gray mice. Now, let's say, for instance, something happens in that population and the dark colored mice have greater fitness. And the lighter colored mice have less fitness. And remember, what we mean by that is, in this particular population that we're talking about, the darker mice have a greater ability to find mates and to reproduce and to have progeny that survive. When that happens, we see a directional selection. So remember, in the time before the selection occurred, the peak of the graph was here. Now what happens is it shifts. And what we will see in this population is, instead of the majority of the mice having a gray color to their coat, we see more mice in that population that have a darker coat color. And we see fewer mice in that population that have a light color to their fur. This could happen for a variety of reasons. Maybe the landscape has changed where it is easier for darker colored mouse to protect itself against predators. Another example would be the finches on Galapagos Islands. And this occurred in 1977 when there was a drought in the islands and surrounding areas. And what they saw over time was a change in beak shape. Finches with a deeper beak, the deeper beak allowed these finches to break open some of the tougher seeds that survived the drought. Prior to that, a more narrow beak was sufficient to survive, but now a deeper beak made it possible for them to eat and get the nutrients they needed in order to survive. And then when they survived, they were able to reproduce more, and then their progeny had deeper beaks. The poor old finch with a more narrow beak, they weren't able to survive as well, and so they weren't able to pass on that trait to their progeny as well. This would be an example of microevolution because it is something that we see within a shorter period of time and is only affecting this one population. One thing I want to make note of as I go through these is I want to talk about whether or not genetic variation has been affected. And so what we see in directional selection is we see a decrease in genetic variation. So in the case of the mice, we used to see a well-distributed coat color within this population, but because of this directional selection, we see many more darker colored mice. So we're losing some of that genetic variation that we used to see. All right, let's move on to stabilizing selection. So again, let's draw the graph that would look like before the selection was applied. And I think it'd be okay if we start off by talking about the mice example that we had in the previous one. Remember, on this extreme we had darker colored mice, and over here we have lighter mice. And in the middle, we have the majority of the mice being represented as maybe a gray-colored mouse, somewhere in between the two extremes. Now remember, with the stabilizing selection, what happens is that we favor a phenotype in the middle, and the extremes become less predominant. So with a stabilizing selection, instead of seeing this nice bell-shaped curve, what we might see is something like this. So the peak is about the same as we see under normal circumstances, but the extremes have been eliminated. We don't see as many darker colored mice or lighter colored mice. Most of them are just this gray color. And again, this could also be for a similar reason. Maybe the gray color has some sort of advantage in that environment. Maybe it provides more camouflage. It protects it somehow. And the darker mice and the lighter mice stand out and maybe more easily captured by predators. So I'm going to just write this here. Average value is the same. With directional selection, the average value, remember, shifted. So average value is the same here. Another good example of this is birth weight in humans. When they study a large population of babies born during a particular time, what they find is the average weight of the baby is around 7 pounds. Going in either extreme of the 7 pounds increases mortality. A baby born that is too small is not going to survive as well. A baby born too big is also not going to survive as well. So those individuals who could have children that were of an average birth weight were more likely to have a child that survived. And we now know that there are genetic factors, inheritable factors, that allow for the variation of size. So those who could pass on this trait of being of an average birth weight were more likely to reproduce, and therefore that trait persisted in the population, whereas the extremes, that didn't persist in the population. As I said on the previous whiteboard when we talked about directional selection, is that we also see a decrease genetic variation. All right, next let's talk about disruptive selection. And we'll start off with our same graph here of number of individuals on the vertical axis, and the horizontal axis is the trait or the allele frequency. So before the selection occurs, we have an average value here, but we also still have the extremes, and we could still think about the darker mice and the lighter mice as an example of darker pigmentation to the fur versus a lighter pigmentation to the fur. And so in this kind of selection, we're going to favor the extremes and not the average. So something like this, where the two extremes have a higher number of individuals with the extreme phenotypes and fewer individuals with this intermediate phenotype. Again, if you're thinking about our mice here, you could imagine a situation that maybe the gray-colored mice, they are less likely to survive because of predators, or maybe they have some other trait associated with their gray coat color that just simply makes them less able to survive, and so they do not pass on the traits as effectively as the lighter colored mice and the darker colored mice. Now, of the 4 different selection modes, disruptive selection is most likely to lead to speciation. Because what happens is we have non-random matings of, say, the light colored mice versus the darker colored mice. And so the longer that goes on, where just the darker colored mice mate with each other and the lighter colored mice mate with each other, the greater chance that each of these, over many generations, could become a new species. Because what happens often is that you end up with some sort of 2 separate populations that only interbreed with themselves. Another example of this would include whitefish of Norwegian lake. These whitefish have structures on them called gill rakers. And I'm going to hijack this graph up here to talk about what the normal distribution would be before disruptive selection occurs. On one extreme, they are long and dense. On the other extreme, they are short and sparse, so not very many of them. These gill rakers are important in capturing their prey. What has happened in this population of whitefish of Norwegian lake is that either the long, dense gill rakers or the short, sparse gill rakers have shown to be advantageous to surviving, more likely to reproduce and to have progeny that survive if they have long, dense gill rakers or short, sparse gill rakers. Those with an average length and average density don't do as well. So why is this? Well, what we believe is happening is that fish with the long, dense gill rakers are more efficient at capturing plankton prey, whereas the whitefish with short, sparse gill rakers are pretty good at capturing small animals. Because their gill rakers are shorter and sparse, they don't get clogged up with mud. Fish with intermediate size gill raker and with an intermediate density of the gill rakers are not very effective at filtering out and capturing plankton, and they're not very effective at capturing small animals. So they don't survive as well, which is why you see a decrease in their numbers. Now, over time, these 2 populations of whitefish with different types of gill rakers in them may lead to 2 different species. With disruptive selection, we say that genetic variation increases. So this is the first one where we see an increase in genetic variation. Okay, let's go ahead and move on to our last one, balancing selection. Now, in this type of selection, remember, no single phenotype is favored in all populations of a species at a given time. So what that means is that the distribution and the average remains the same. These are supposed to be the same. They're not identical, but the idea is that they're supposed to be the exact same. There are a few ways that we can see balancing selection, but I want to talk about 2 specific ways. The first is heterozygote advantage. And what this one means is that heterozygotes have greater fitness than homozygous individuals. We haven't mentioned the words heterozygous or homozygous, but let me do that real quick. Remember, as we said before, every individual has 2 alleles for any given gene. And so let's use something simple and say the allele could be A. So you could have 2 alleles that are big A, big A. You could also have 2 alleles that are little a as the 2 extremes, and then the one in the middle is big A, little a. The prefix homo means the same. So what we're saying is they have 2 of the same alleles. So big A is homozygous, little a are also homozygous. Big A, little a hetero means different. So they have different alleles, big A, little a. So these would be heterozygous individuals. So what we're saying about heterozygous advantage is that for some reason, heterozygotes have some sort of fitness advantage over these two extremes, these two homozygous individuals. So let's talk about an example of heterozygote advantage. Classic example of heterozygous advantage is looking at the diseases of sickle cell anemia and malaria. Sickle cell anemia is a disease of the red blood cells. Instead of forming a nice circular cell, a nice flexible cell, it forms this rigid sickle-shaped red blood cell that doesn't transport oxygen very well and can get clogged up in our blood vessels and can lead to death. Malaria is a disease transmitted by mosquitoes to humans, and it can also lead to death. Now, interestingly, when we look at the incidence of sickle cell anemia and malaria, we find some overlap in the continent of Africa. We see regions that have higher incidences of sickle cell anemia that overlap with regions where malaria is the highest. So why is that? Well, when we look at these two diseases, we see some genetic differences here. So if we look at sickle cell disease, which I'm just going to put as SCA for sickle cell anemia, we know there are three combinations, and we're just going to call them big S, big S, big S, and little s, little s. People who have big S, they do not have sickle cell anemia. People who are big S, they also do not have sickle cell anemia. Individuals who are little s, they do have sickle cell anemia. Interestingly, what they discovered was that there is a difference in malaria susceptibility depending upon what your genotype is of the sickle cell disease, of the sickle cell genes. So here let's put malaria, I'm just going to put MAL, resistance. People who are big S, they are not resistant to malaria. If a mosquito bites them that can transmit malaria, they will get malaria. Those who are little s, they are resistant to malaria. However, this is not a good trade-off. You have sickle cell disease, but you won't get malaria. So that's not necessarily what you want. Interestingly, individuals who are heterozygous for the sickle cell mutation, they are also resistant to malaria. So this is where the advantage to being a heterozygote comes into play. Being heterozygous for the sickle cell gene, they won't have malaria, but they have this added benefit of also having resistance to malaria. Now, because of this, these individuals who are big S, little s, don't have sickle cell anemia, don't get malaria, so they are more likely to pass on their genes to progeny. And by passing on these genes to their progeny, you're more likely to have more individuals in a population who will be resistant to malaria. So this kind of selection pattern selected for the heterozygote because they had a better chance of surviving. You still see people with big S, and little s being produced, and they have some disadvantages of getting malaria or getting sickle cell disease. But the heterozygote, they have neither of those problems. The second example of balancing selection occurs when there is something we call frequency-dependent selection. So we see frequency-dependent selection when certain alleles are favored when they are rare, but not necessarily when they are common. A good example of this is seen with lizards found in the Pacific Northwest, and we're going to call them side-blotched lizards. Now, males of the side-blotched lizards have three different colors to their throat. They are either orange, blue, or yellow. And I don't have a yellow marker, so I'm going to use pink, but the color is yellow. Now, each of these has a different strategy, a different reproductive strategy, and all three of them are successful in this strategy. Sometimes orange lizards are more successful, sometimes blue ones are, and sometimes yellow ones are. It's kind of like an elaborate biological game of rock, paper, scissors. Let me explain what happens here. Okay, so here is a picture of the orange, blue, and yellow side-blotched lizards, and these are males because we see this color pattern only in the males. And remember, I said that each one of these has a different strategy for reproductive success. The orange males, they're the strongest, and so they outcompete blue males. In our rock, paper, scissors analogy, orange beats blue. The blue males, they're of a medium size. They're not going to be able to take on an orange lizard, but they can guard off the yellow lizards. And that's because they have a stronger mate bond. They're more likely to be near the nest or the female, so they're able to prevent the yellow lizards from gaining access to the females. And remember, I said I didn't have a yellow marker, so I'm going to use pink to talk about the yellow males. They're the smallest, but they're the sneakiest. So remember, orange beats blue, blue beats yellow. What does yellow beat? It turns out yellow beats orange in this game of rock, paper, scissors. And that's because they sneak in and they can mate with the females that the orange males would have mated with, because the orange males don't have this stronger mate bond connection. So that gives the yellow lizards an opportunity to sneak in and reproduce with the females. So let's say they're sneaky. Orange beats blue, blue beats yellow, yellow beats orange. Because of this, we see a frequency that is over time that is pretty well distributed. However, the short-term frequencies of the different alleles that lead to orange, blue, or yellow can vary. Let me show you what I mean by that. So we have number of individuals on the vertical axis, right? Still, that hasn't changed. And here what we're going to call this is traits over time. The orange ones, sometimes they're going to be a higher frequency and other times they'll be lower. You might go back up. The blue ones are probably not going to follow this exact same pattern, but rather maybe they'll be something like this. And the yellow ones will be something like this. Where you see in this graph over time, times when there's more blue lizards, times when there's more yellow lizards, and times when there's more orange lizards. But over time it balances out. And in this case, we would say that genetic variation is maintained. Okay, let's move on to our next objective, and that is to talk about sexual selection. Initially, Darwin was perplexed as to why males in some species were more brightly colored or more elaborately colored than the females in that species. At first, he didn't see what the adaptive advantage would be to putting all that energy into producing that elaborate coloration. So, for instance, we see in this picture the male peacock and the female peahen. So why is it that the male would spend so much energy to producing this beautiful coloration of plumage? And why wouldn't the female? That's a lot of energy to make all of this. He reasoned that the adaptive advantage was that the more brightly colored the peacocks were, the more likely they were able to attract a mate. And if they were able to attract more mates, then they would have reproductive success. This became known as the fundamental asymmetry of sex. It is also referred to as the Bateman-Trivers hypothesis. And this is based upon the observed pattern that we just talked about. And that is that traits that attract the opposite sex are more elaborate in males. Now, of course, not in every species, but in many species we do see this, just like in the peacock and the peahen example. This led to a hypothesized process that could help explain this observation. This hypothesis states that eggs are more energetically costly than sperm. Therefore, females invest more in their offspring than do males. An egg is much larger than a sperm. It also is packed full of nutrients and material that is necessary to sustain life once it is fertilized. The sperm in itself is just a carrier of genes to be delivered to the egg. While there is some energetic cost in making sperm, it is not nearly as much as it is in making eggs. So females put a lot more energy into making eggs than males, and that allows males to spend more energy on these elaborate traits. Another way to think of this is from the viewpoint of fitness. So female fitness is limited by the ability to gain resources needed to produce eggs. So the food and energy she uses has to go to making high quality eggs. So in the term of fitness, if she can make these high quality eggs, then she will be able to pass on her genes more effectively than a female who cannot make as high quality of eggs. On the other hand, let's think about male fitness. It's limited by the number of females he can mate with. So he wants to put more energy into things that will attract more females, or maybe more energy into ways to outcompete other males. But his fitness, his ability to pass on his genes, is really a matter of how many females he can mate with. So the Bateman-Trivers hypothesis makes a few predictions. The first is that females, but not necessarily males, should be choosy about mates. Since they invest a lot of energy in each egg. Similarly, males should be willing to mate with almost any female, because they're not spending a lot of energy in the production of their sperm. This is called intersexual selection. Because one sex, females, is being choosy about which males to reproduce with. So it's called intersexual. The second is if there are equal numbers of males and females, or roughly equal numbers, then males will have to compete with other males for female mates. This is called intrasexual selection. The third prediction is that alleles that increase a male's attractiveness or success in male-to-male competitions should increase in a population. So that's why we would see more brightly colored peacocks than dull colored peacocks, because those brightly colored peacocks had a greater probability of reproductive success, and therefore the alleles that cause brightly colored feathers, those are going to become more predominant in a population because they represent reproductive success. So now the question is, when we look at natural populations, do these three predictions hold up? Is this what we actually see? And the answer is sometimes. In some species, in some populations, those three predictions are supported. However, there are always exceptions. And just briefly, some of those exceptions are sometimes we see males investing more energy into the offsprings. females in some species benefit from being promiscuous. That is, having multiple sexual partners. And sometimes we see where males benefit from being monogamous. That is, having a single sexual partner. Other factors also have to be considered. Things like availability of mates, food resources, and other factors. Like most things, when we look at the final explanation, there are many factors that can influence what actually happens in a natural context. But the predictions of the Bateman-Trivers hypothesis do hold up, as I said, in many different kinds of species populations. So now let's continue this discussion and talk a little bit more about intersexual selection. Remember, that is when females in a particular population are choosy about which males they mate with. So what traits are they looking for when they're being choosy? We know there are physiological or behavioral traits or physical traits that the female may look for in the male. But what is it about those traits that tell the female that this is a good male to mate with? And there's this hypothesis that we call it the honest signal hypothesis. And what this hypothesis states is that these honest signals, they state something about the health and genetic quality of the male. Let me spend some time talking about an example that supports this hypothesis. In some bird populations, males are bright red and yellow. Red and yellow pigments that cause the red and yellow color are made from carotenoids. We know that carotenoids protect tissues and stimulate the immune system. To obtain carotenoids, the birds have to eat carotenoid-rich plants. Okay, I'm going to erase this because I ran out of room, but we're going to pick up on this in just a moment. So from the female bird's perspective, bright red equals a healthy and well-nourished male. So the prediction then is that there are what we might call good alleles that contribute to this pigmentation. It seems reasonable that there would be some alleles that would allow males to be better at getting these carotenoids to show to the females that they are healthy and well nourished. Okay, let's go ahead and talk about intersexual selection. In some species, we see male-to-male competition as the primary cause of sexual selection. A good example of this is looking at elephant seal populations. In these populations, males are much larger than the females. The males can be upwards of 6,000 pounds. I'm not going to ask you on a quiz or test how much they weigh, but that's just to give you a perspective. These elephant seals, the males are huge. And these males, they will fight each other for mating territories. The female elephant seal populations, they come on shore for the mating seasons. And so males fight vigorously against each other to establish an area that they claim as their own territory. keeping all other males away. And then when the females come on shore, they have shown that they have a prime mating area, so to speak. So often size and fighting strength is what will win the day. I suspect I would not have been very successful as a male sea lion. So in this example, it's all about fighting for and maintaining that territory for mating when the female elephant seals come ashore for mating. So the last thing I want to talk about in this podcast is something called sexual dimorphism. So we've talked about this throughout most of this video podcast. We just didn't give it a name. So let me define what sexual dimorphism is. These are any trait that differs between males and females of the same species. These can be a wide variety of things. They could be some kind of weaponry or defense, ornamentation, like we see with the peacock behaviors. Some males in some species will have an elaborate courtship dance, and body size, just like we talked about just a moment ago about the elephant seals. As we said before, males typically will have more traits that function in courtship or competition for mates than females will. All of these, and others that I didn't list here, lead to reproductive success. Either because the male can outcompete another male, or because these traits signal to the female to mate with the male, or because these traits signal to the female to mate with the male. Another important thing to remember is that these sexual dimorphic traits are heritable. Or another way of saying it, there are alleles responsible for these traits. All right, in this video podcast, we covered a lot of material. We began by picking up something that should have been probably on the last podcast, but it fits in here well just the same. And we talked about the difference between macro and micro evolution. How macro was on a much larger scale that would only be seen over a long period of time, like millions of years. And micro is something that would have been seen over a much shorter period of time and is seen within individual species and populations. We spent a little time talking about artificial selection. We talked about the modes of selection and how that influence genetic variability. And we just ended with talking about sexual dimorphism. All right, that is all I have for this video podcast. If you have any questions at all, please make sure you reach out to me and I'll help you as best I can. If not, I will see you in class. Bye for now.